everybody. So the topic of today's class is random matrices or random networks. So we concluded on Thursday. So we did two things toward the end of class on Thursday. First, we discussed the real distribution of synaptic efficacies at least in the cortex. And I told you that there is evidence, although not everybody accepts it, that the distribution of synaptic efficacies is log normally distributed. We also discussed the Hopfield model. We went back to the Hopfield model, and we concluded that in the Hopfield model, the distribution of synaptic efficacies is normal, but the efficacies of different synapses are not independent. First, there is symmetry. Jij is equal to Ji, and we have good reasons to believe that this should have a large effect on behavior, because we have this theorem that tells us that in symmetric networks, the dynamics will converge to a fixed point. And second, because there is structure. There is structure. I see that I'm running out of battery. There is structure in the sense that the rank of the connectivity matrix is relatively low. Would be in general low. So what we will do today, we will very briefly discuss the dynamics when connectivity is random, going beyond the Hopfield model. And we will start by considering symmetric networks. So the general equation that we always write is Ri dot is equal to minus Ri plus hyperbolic tangent of beta sum over j of Jij Rj. These are the dynamic equations as always. And we will consider the case in which Jij is equal to Ji, and it is normally distributed between from a normal distribution with a mean of G square over n. And the question: what do we expect the behavior of this? What's the expected behavior of this network? So, well, if beta is equal to 0, then it's easy. Everything will converge to 0. And interesting things can happen if beta is sufficiently large. Now, in order so imagine that this is an exam and you're faced with this question. So what would you do? What will happen to the dynamics? Converge to what? Which fixed point? Okay, good. You know that it's symmetric. So you know that it will converge to a fixed point. But the question is, which fixed point? What are the fixed points? I'm sorry? Well, okay, so we can ask the question, what will happen in different limits of beta? For example, if beta is equal to 0, that's easy. Okay. If beta is very large, it's more difficult. So let's assume that beta is non-trivial. It's not 0. What are the fixed points? At least one fixed point you can find, right? the 0 fixed point. If r is equal to 0, then this is equal to 0, and this is equal to 0, and r dot is equal to 0. So there's one fixed point which is the 0 fixed point. And if we start with beta that is equal to 0, and we slowly increase beta, maybe this fixed point will become unstable. So one question worthwhile asking is, what are the conditions such that the 0 fixed point is, the 0 homogeneous fixed point is stable? So what do we need to do in order to address this question? Linearize. Okay. Let's linearize it. So the linear equation will be delta r I dot is equal to minus delta r I plus beta sum over j of jij delta rj, where we linearize around ri is equal to 0 for all I. Okay, so we have a set of linear equations. Okay, next. What should we do next? What do we do when we have a set of linear equations? I'm sorry? So we need to find the eigenvalues of this matrix J. So if we know the eigenvalues of this matrix J, then we will know the eigenvalues of the matrix beta times J. Let's write it in a delta r dot is equal to minus I minus beta j delta r, writing it in using matrix notations. So if we know the eigenvalues of j, then we know the eigenvalues of this term, and we will be interested in finding the largest eigenvalue of j. So the question that we are interested in is what will be the distribution? So in general, we cannot compute the eigenvalues of J because J is a stochastic matrix. But we can say something about the distribution of eigenvalues. Maybe we can say something about the distribution of eigenvalues. So one thing that we can do is we can use our intuitions from the Hopfield model, because in the Hopfield model, Jijs are normally distributed. So in the Hopfield model, what is the value of G? So it will be, well, G square will be, G square will be equal to P over N in the Hopfield model. Now, in the Hopfield model, when will the 0 fixed point lose its stability as a function of P? So let's say that P is equal to 1. What are the values such that the fixed point will lose its stability? If P is equal to 1. the number of memory patterns is equal to 1, when will the zero fixed point become unstable as a function of beta? When beta is equal to 1, then the homogeneous fixed point will lose its stability, or for beta larger than 1, the homogeneous fixed point will become unstable. So using this intuition, we might expect that it's sufficient for G, let's say that for the sake of simplicity, let's assume that beta is equal to 1. For beta being equal to 1, the Hopfield intuition is that G that is 1 over square root of n is sufficient for instability. Right? So this is the intuition that we have from the Hopfield model. But it turns out that this intuition is wrong. Turns out that this intuition is wrong because the connectivity matrix in the Hopfield model is very non-typical. The connectivity matrix in the case of P is equal to 1 is P transpose. This is a connectivity matrix in the case of in the Hopfield model with P being equal to 1. And this is very different from randomly choosing J's from this distribution. And well, okay, so we don't know what the structure of this what's the distribution of eigenvalues. So we can simulate it. Show you how it looks in simulations. So we can start with we can start with a small network. So this is a distribution of the eigenvalues when G is equal to 1 in the case of a small network with n neurons. And every time that I will run it, I will get different numbers. So this is how the distribution looks like. But you can see that it goes something like between -2 and +2. This is for G is equal to 1. We can consider a larger network. And for a larger network, every time that I run it, I get a different realization, but they're not too different from each other. We can go for larger. And despite the fact that every time I have a different realization of connections, they are not so dissimilar from each other. I'm taking an even larger network, 500 neurons. Or 1,000 neurons. You can see that there is a rather typical structure. 2,000. So the eigenvalues go from -2 to +2, and the shape is approximately, well, it's a semicircle. The distribution of eigenvalues approximately a semicircle, and it turns out that this is not a coincidence. There's something that is called Wigner's semicircular law. There's something called Wigner's semicircle law, and basically tells us that if J is equal to Ji is normally distributed around 0 with variance that is equal to G square over n, in the limit of n going to infinity, the distribution of eigenvalues, or the eigenvalues are distributed in a semicircle between -2g and +2g. So going back to our distribution, and you see that it's around semicircle between -2 and +2. This is because we chose g to be equal to 1. It turns out that in the limit of n going to infinity, this is a general case. So this tells us when conditions under which the zero fixed point will lose its stability and will become unstable. Questions? So another question that one may want to ask is, okay, so we know that if beta times 2g exceeds 1, then the zero fixed point will become unstable. We know from our theorem that dynamics will converge to a fixed point. The question is, well, can we say anything else interesting about these fixed points? For example, what will happen in the limit of very large beta? So we can go back to our Hopfield model intuition. So in the Hopfield model, let's try it again, the Hopfield model. So in the Hopfield model, so Jij in the Hopfield model is equal to 1 over n sum over mu. Let's write it let's use a matrix notation, p mu transpose. And we discussed the fact that the rank of this matrix, if p is small, if the number of memory patterns is small, the rank of this matrix is small. as P increases, the rank of this matrix increases. So one way of thinking about it is that the larger P, the larger the number of memory patterns, the more similar does the Hopfield model become to this model. So in the limit of very large P, the Hopfield model becomes similar or converges to this model. Because the correlations in the connectivity disappear. Now, we already know something about the structure of fixed points in the Hopfield model and how this structure or number of fixed points depends on the number of memory patterns. So what do we know from the Hopfield model? What are the fixed points in the Hopfield model in the limit of large beta? The patterns themselves are fixed points, okay. And mixtures of the patterns, odd mixtures of the patterns. So we studied analytically the case of 3 memory patterns, but from the analysis it was clear that if the number of patterns is larger, then the intuition will hold for 5, mixtures of 5, mixtures of 7, mixtures of 9, mixtures of 11, and so on. Now, the number of mixtures is clearly much larger than the number of actual memory patterns. So we expect that in this model, in the limit of large beta, the number of fixed points will be very large. And it turns out that indeed this is the case. So in the limit of when beta goes to infinity, the number of fixed points of the dynamics is exponential in the size of the network. So let's say it again. The number of fixed points is an exponential function of the number of neurons in the network. So the total number of states in the limit of beta going to infinity, the total number of states in the network would be 2 to the n. It's an exponential number. It's a number that is exponential in the number of neurons in the network. Similarly, the number of fixed points, not all of these 2 to the n, not all of these 2 to the n states are of course fixed points, but the number of fixed points will also be an exponential number, will be exponential function of the number of neurons in the network. In physics, this is known as a spin glass state. And in fact, a lot of research on the dynamics of neuronal networks, much of it is based on similar work that was done in physics. Questions? Everything's clear? Okay, so the next question that we may want to ask ourselves is, what will happen if connectivity is random and non-symmetric? We don't need this. This. So what should we expect if the Jij's are normally distributed, but this time without any symmetry. Now, again, a way to address this question would be to linearize the equation and to ask the question, when will the zero fixed point become unstable? This is one question that the first question that we would like to ask, and this would depend on the eigenvalues of this matrix J, but this time we're not talking about a symmetric J, but a general J. Now, going back to simulations, we can do the same trick, just simulate it. So what I'm plotting here is it's kind of difficult to see. Every point here is one eigenvalue. This is a real part of the eigenvalue, and this is the imaginary part of the eigenvalue. So when the matrix is symmetric, then we know that all eigenvalues are real. I'm sorry? Yeah.
You just amortize the
Better? So this time the eigenvalues will be complex. In general, will be complex. So this is the real part, and this is the imaginary part. And every time that I simulate it, we get a different eigenvalues. But it turns out that in the limit of large n, the distribution, there's structure in the distribution of eigenvalues. So now I'm going to a network with 100 neurons. How many eigenvalues will there be? The matrix is n by n. So there'll be 100 eigenvalues. You can already see that there's some structure. $200. $500. $1,000. $2,000. Well, it seems that they are all uniformly distributed in a circle. And it turns out that this is really true. And as always, there is a Russian that proved it. So why don't I go beyond 2000? Because then it takes too much time. Just a matter of time and memory. And we have a law that is called Girkov's circular law. Girkov's circular law that tells us that if Jij is normally distributed around 0 with the variance of G squared over n, then again, this is supposed to be a circle. So this is real. This is the real part of lambda, and this is the imaginary part of lambda. And what Girkow's circular law tells us that the eigenvalues will be uniformly distributed in this circle. Okay, so in the limit of n going to infinity, the eigenvalues will be uniformly distributed in this circle. And clearly what we care about is which eigenvalues. What do you mean by largest? We are interested in the eigenvalues whose real value is maximal, which would be this one. So we know that the real part of the largest, the eigenvalue that has the largest, the real part of eigenvalue is will be G in this case. So we know that the zero fixed point is stable as long as beta G is smaller than 1, and when beta G is larger than 1, then this zero fixed point will become unstable. So now the question is, okay, it will become unstable, but what's going to happen? In the case, in the symmetric case, we know that there will just be more and more fixed points, and in the limit of beta being large, then there will be then the number of fixed points will be exponential. But the question, but now we're interested in this problem where we don't have this theorem that tells us that things will converge to a fixed point. So the question, what will be the behavior of the network in this case? Now, before telling you about the behavior of the network, one may want to ask about neurons in the brain. What can we say about the connectivity in the brain? Is it symmetric? Is it not symmetric? Last week we concluded that, or I told you that the distribution of connections in the brain is not normal. It is log normal, or maybe log normal. So one may want to know what will happen if we will consider a different distribution. Now, it turns out that as always, many of the results that we obtain for simplified toy models are also applicable for more complex networks. So with respect to the brain, there's the issue of neurons being either excitatory or inhibitory. This is a major difference from what we have here. And there's also an issue of motifs, and this is something that is being extensively studied these days. The idea is that if neuron A is connected to neuron B, it's more likely that neuron B will be connected back to neuron A. It's even more likely, even when controlled for the distance. So networks are not completely random. There's structure in them, but nevertheless there's a lot of heterogeneity and a lot of what seems to be like randomness. So this is talking about the brain. But going back to this problem, this is a well-defined problem, and we can ask ourselves, okay, what will be the behavior? Now it turns out that if beta is large, then the behavior of these networks, the behavior is chaotic. And I will show you using a simple simulation that Itamar wrote. So I'm going to consider a network with 800 neurons with beta that G is equal to 1 and beta is equal to 3. So let's talk about the structure of the eigenvalues. Sorry. So this is a distribution of eigenvalues, and this is the prediction from Girkov's circular law. Again, the prediction is only true in the limit of large n, but you can already see that for 800 neurons, the relation of the theory to the actual simulation is pretty close. Now we can talk about behavior. So what you see here in black, you see the average activity in the network, and these three colors denote the activities of 3 randomly selected neurons. And it's pretty messy. The reason that it's messy is that the dynamics are chaotic. So the way this property of asymmetric networks is that they yield dynamics. They yield chaotic dynamics. Another way of plotting it is to look at the activity in 3D space, activity of, again, 3 randomly connected neurons, and, well, now this is not a proof, of course, that the dynamics are chaotic. It could be periodic. The issue of chaos is rather complicated. I don't remember if you had did you have an exercise about Lorentz and chaos and the Lyapunov exponent? I'm sorry? I'm sorry? Okay. So we can look at the dynamics when considering two starting from nearby initial conditions, how the dynamics changes starting from nearby initial conditions, and this is the behavior, and this line is the exponential fit, enabling us to compute the Lyapunov exponent. We can study the effect of perturbation. What happens if we, at some point in time, we just slightly perturb the activity? So you can see how the specific trajectories of the neurons they diverge from the original the average activity, how they changes because the average activity is very close to zero. We can look at it again, looking in the space of these three neurons. So you can see how the trajectories diverge. The blue is perturbed and the red is original, and at some point they become completely uncorrelated. So the conclusion from these results is that the rate equations when connectivity is symmetric is yield dynamics that are very different from what we are used to, from these fixed points. Rather than having fixed points or simple limit cycles, we have these networks exhibit a rich repertoire of behaviors, and in the right conditions these networks are chaotic. Questions? Yes?
So can it be useful in order to describe neural activity if there is no big scale? Or should we stick with the symmetric arrangement?
Well, neural activity is something very rich. You can't just say neural activity. We have the different cortical areas, you have different conditions, different regions. When you look at the first recordings of neural activity were EEG, and there you have very rich behavior. Sometimes you see very nice oscillation, sometimes it looks more chaotic. So this is we have our theoretical tools that tell us how the connectivity will affect activity. And we have different regimes. There are regimes in which we have a particular computation, like the regime of a ring model, or when we discussed about line attractor. This is one regime. There's the Hopfield regime. There's the symmetric networks regime, and these random networks regime. Now, the true connectivity in the brain is neither. But what we learn from these models is how changes to the connectivity are expected to affect the activity. So the question, the relevance to the brain, this is too broad a question. Once you will study your own favorite brain area, you will be able to say whether the activities, whether the tools of the Hopfield or the tools of Ring or these tools are relevant to describing activity there. So you can think about it as a methods course. It can be different methods or different frameworks are applicable to different brain regions or different conditions. More questions? So if there are no more questions, we will take a break now, and after the break we will move away, and maybe it's also relevant to your question, we'll move away from talking about rate. All these models are models of rate, and we will start considering the individual action potential.
בגדול מה שאנחנו עושים. אפשר רק להגיד, זה כל מיני סיבות בחירה, זה ימנן, כי היה גם כאלה שהיה שעבור בנקור אותו, עשינו 13 לקורס, זה, ולא ניגש למבחן, בלגן. עשיתי 13 לקורס, שוב בית לבוא לפי כל שנה גיל. אתה כמה כאלה?
So far we talked about rates, but these rates are made of spikes. And whenever we look at neural activity in the cortex, it looks like a mess. So this is an example, classical example from the work of Georgopoulos, and he trained monkeys to move a manipulandum to one of 8 directions. And what you see here is a firing rate as a function of direction. These are rasters of individual trials, and the firing rate is a function of the direction. And we talked about this type of tuning. And but when you look at individual trials, there are 2 things that are worthwhile noting. One is that the firing rate or the timing of spikes within an individual trial, the timing of spike is rather irregular. So in this example, there were in this neuron fired 2 spikes, and then there was a long period of no activity, and then higher rate, and then nothing, and so on and so forth. So this is one thing that is worthwhile noting. A second thing, which I find more disturbing, if you like, is the fact that if you consider different trials that compare this trial to this trial or this trial to this trial, the pattern of activity is very, the timing of individual spikes, this timing differs between trials. Now, I think this is horrible. This is horrible in a very deep sense. Now, when we do experiments, when we repeat an experiment, we expect that whatever we measure will also repeat. I mean, this is science, right? You should be able to do the same experiment again and again and get the same results again and again, but we cannot. The question is why? So why can't we do it? There is noise. So the standard answer is noise. And Hamehadrin, I don't know how to translate it to English, would say biological noise, right? There's noise in biology, right? Okay, so if there is noise in biology, what would be the prediction of this hypothesis that this is due to noise in biology? I'll tell you what I would predict. This is a kind of answer that I hate. I'll tell you why. I hate it because it's cowardness to say noise. Because if you say noise, then it implies that, okay, this is beyond me. There's nothing I can do about it. I just have to live with it. And since we are scientists and we really want consecutive trials to result in the same response. So, but you say noise, and this is what people typically say. So one prediction, I would say, of this idea of noise would be that if you do the experiment more carefully, the magnitude of this noise should be reduced. This is, I would say, a prediction of this claim that it's due to noise. Now, this is an experiment from 1982. Judging from the year, it's like 1 million years ago, right? Since then, everything has changed. This is a newer study. This is from this is a review from 2011. Monkey is doing exactly the same experiment, and these are the spikes before movement, and this is movement onset, and it is still as noisy as it was in 1982. So this noise thing is something that we are unable. Well, first, this is something that has been there for many years in our experiment, and it doesn't seem to be something that we were able to get rid of, despite the fact that noise sounds like something that if you do the experiment more carefully, then you'd be able to reduce it. Well, we are unable to reduce it.
But there is noise in the system. Noise doesn't have to be from the experiment.
What do you mean by noise in the system?
Things are probabilistic. I mean,
Why should things be probabilistic? What are the things that are probabilistic in the system?
if the neuron is higher given specific input,
So you argue, if I understand you correctly, you say, well, the reason for what seems to be noise are stochastic processes taking place within the individual neuron, these processes that underlie the firing of action potentials, right? That would be the claim. Now we can address this question in two, or we can address this hypothesis. If this is indeed the case, then this result in some sense will not be surprising. Now we can address this question in two ways. We can address it theoretically and ask, is it likely, what are the stochastic elements in the neuron, and can these stochastic elements underlie this behavior? This is one way that we can address it. And another way would be to do experiments on neurons and ask, do neurons, if we carefully manipulate individual neurons, not the animal, but individual neurons, do we see this level of stochasticity? Right? So let's start with your first point. Let's start with the first point, which is the neurons are stochastic. So why should neurons be stochastic? What kinds of what in a neuron can be stochastic? Channels. So what are channels? Channels are proteins, and they have a structure. And the difference between an open channel and a closed channel is the structure. And if we measure individual channels, indeed we see that channels open and close in a way that could be interpreted as stochastic. And there are even reasons why this should be stochastic, and this would be because at the level of individual molecules, there is stochasticity that emerges from the stochasticity of physics, from quantum mechanics. So this is one place where stochasticity could emerge and perhaps could yield this behavior. What other aspects in a neuron can be stochastic or are stochastic? Vesicle release, right? So communication between neurons, the communication itself is stochastic. Now, putting aside the mechanisms underlying it, we know that an action, the response of synapse to an action potential differs between consecutive action potentials. And again, the model that we use for synaptic transmission, these models have a stochastic component in them. So both these mechanisms, in principle, can underlie this behavior. On the other hand, there are reasons why they should not. For example, if we think about a cortical neuron, how many synapses, how many inputs does a cortical neuron receive? Approximately 10,000, few thousand at least. So even if there is stochasticity in individual synapses, we expect this stochasticity to average out when considering a large number of synapses. Similarly, there is a very large number of ionic channels. So even if there is stochasticity at the level of an individual ionic channel, this stochasticity should average out if there are enough channels. So now the question becomes a quantitative question. Is stochasticity at the level of synaptic release, is it sufficient? Or is stochasticity at the level of channels, is it sufficient relative to the number of channels? So rather than addressing this question theoretically, we can do experiment in individual and study the extent to which individual neurons are stochastic. And it turns out, and I don't know if I have slides here, it turns out that at least in a slice, there's very little stochasticity. So despite the fact that in principle stochasticity at the level of channels could have underlied this, we don't see it in cultures. So it doesn't seem to be an explanation. Just more examples. This is a motto. This is again going back to the 1980s. These are experiments in the temporal cortex in which a monkey is trained to memorize a pattern, and these are repetitions of the same pattern, and again activity is stochastic. This is area LIP. I will not go into the details of what is studied here. People are interested in these averages. But again, if we look at the individual neurons, there's a substantial volatility. This is an example from the cerebellum, just to show you that neurons can be very regular. This is an interneuron in the cerebellar cortex. This is in vivo. So all the noise that you are talking about, telling me, okay, biological noise, whatever, you don't see it here. It's like a pacemaker. I know people don't care about the cerebellum, but nevertheless, it's not necessary that a neuron will be stochastic. There's nothing, if you look at the synapses, if you look at the channels of this neuron, there's nothing that's obviously different from cortical neurons. Again, could be that some details were missing, but there's nothing here. This is an interneuron in the cortex. Well, not in the cortex. This is a slice. And this is a response of a neuron to current injection. Different levels of current injection, like a clock. But this is current injection, so it could be that it's all the stochasticity of synapses, at least. The mismatch between this result and in vivo results. But there is a puzzle here that is worthwhile addressing. There's an interesting puzzle here. Now, before addressing this puzzle, I'm going to make a rather long detour and discuss the building blocks necessary for addressing this puzzle. And there will be two building blocks. One is I will describe the way of characterizing this irregularity. And then I will describe a simple model for the generation of action potentials. And we will use these two tools in order to go back and ask how come that activity in the cortex is first irregular and second not exactly reproducible. And it will turn out that this is not a noise in a simple sense. It will turn out that this is something that will naturally emerge from the model, this kind of behavior. Questions? So if there are no questions, I will start by telling you something about point processes. So, when we look at a raster plot, what we care about is the timing of the action potentials. So it's a process where we care about the time of things, and these kinds of processes are known as point processes. So in neuroscience, we care about the timing of action potentials. Maybe you care about I don't know, if you wait in a bus station, you care about the timing of when will a bus arrive. Don't care about the color of the bus or the gender of the driver. All you care about is when will a bus arrive. I don't know, if you work in a customer service, you care about the timing of phone calls. When will someone make a phone call? And we will restrict ourselves to analyzing situations in which only a single event can happen at any point in time. And we would like to define instantaneous rate of events, and we will we'll denote it by lambda of t. Lambda of t will be the rate of events, such that the number of events between t1 and t2 will be an integral from t1 to t2 of lambda dt prime dt prime. We don't need a prime. Lambda t dt. So this is our instantaneous rate, and the number of events will be just the sum, the integral over the rate. So we can think about lambda of t as a sum of delta functions, where ti's will be the times of these events that we're interested in. For concreteness, we'll think about action potentials. Now, we will consider stochastic processes. So these point processes can be, or we will think about them as stochastic processes, and then we can think about things like the average of lambda or the variance of lambda. So the interpretation So the way to think about about the average of lambda is to think about, so let's say that we repeat the same experiment again and again and again. So we repeat the experiment again and again and again, and this is what we get in our experiment. And we think about a particular point in time. Let's say this point in time t, and so lambda of t would be the average number of events occurring around this t. So basically, or more formally, so lambda of t will be the limit of delta t going to 0. So this will be delta t. This interval is delta t. And the number of experiments will take the number of experiments to go to infinity. So we'll take this limit to the limit of the interval to 0 and the limit of number of experiments to infinity, and we will just count the number of events between t in the interval of t minus delta t over 2 to t plus delta t over 2. So we'll count the number of intervals occurring in this line and divide by the number of experiments and this interval. So lambda of t, the average of lambda of t, will be taking an infinite number of experiments, delta t going to 0, counting the number of events occurring here, normalizing by the number of experiments and by delta t. Okay? So this would be the meaning of the average of this instantaneous rate. Questions? So another way of thinking about point processes, rather than looking at the times of the individual events, we can think about the intervals. We can think about these intervals. So I'm going to define capital T to be the interval, and Tk will be defined as the k plus 1, the timing of the k plus 1 event minus the timing of the k event. Now, if Tk is the timing of these intervals are independently and identically distributed, if the distribution of Tk are IID distributed, then the sequence of times called a renewal process. So that's another way of thinking about it. So we can talk about the timing of the individual events, or we can talk about the intervals between the events. So one process which is very useful is known as Poisson process. So in Poisson process, what we do, we take our time and we divide it into intervals of size delta t. And in a Poisson process, the probability of an event at t minus delta t over 2 and t plus delta t over 2 is equal to lambda t times dt. So there is some probability that there is an event at an interval, and we will take the limit of delta t to 0. So this is a Poisson process, and a homogeneous Poisson process. Homogeneous Poisson process is lambda of t is constant in time. So this is very useful for a homogeneous Poisson process. The concept of a Poisson process is very useful, and specifically a homogeneous Poisson process, and therefore we will devote some time studying homogeneous Poisson processes and their properties. Questions? What would be an example of a homogeneous Poisson process? The regular cell, you mean? No. Well, no, the cell will not have this property. Why is that? So the cell in the cerebellum had this, looked like this. Right? That's the spike count of a cell. So clearly, well, if it fired here, it will not fire here. We take the limit, the delta t to 0. Which neuron? Well, you said baseline neuron.
That's not firing at all.
Neuron that doesn't fire at all. Okay, a neuron that doesn't fire at all is okay. I could be. I need to think about it. It could be a Poisson process, but it is certainly not an interesting Poisson process.
Why not the Drosophilus?
I'm sorry?
The Drosophilus case?
Well, how would you know?
You're not sure. No.
What would be a signature? Okay, I agree that it looked like a mess. The example with the bus. So the example with the bus, is it an example of a Poisson process? Well, it depends on the bus company, I would say.
Nothing is.
Well, actually, maybe it isn't. I'm sorry? Exactly. So the idea is that you have many buses, and each come at its own rate independently of the other. So I don't know if this is an example. Well, vesicle release, again, this would be a question that we will need to address experimentally, and we will discuss the signature of a Poisson process. But just to give you an insight, so assume that you are you're working as you're receiving calls, you're in a customer service place, and you have people from all over the world calling you because they have an issue with I don't know, with the software that your company is selling. Now, assuming that there's nothing external that affected all the software, then different people will call at different times independently of each other. This would be an example of a Poisson process. Now, this could be homogeneous or non-homogeneous. For example, you could imagine that during the day more people call than during the night. And this would be an example of a non-homogeneous Poisson process. We will soon see several examples of a Poisson process, both relevant to neuroscience and irrelevant to neuroscience. But some of you suggested, for example, that vesicle release is a Poisson process, or that this Georgopoulos neuron is a Poisson process. Now, in order to be able to address this question, we need to study what the signature, what's experimental signature of a Poisson process. So this is what we will do now. So the question that one would like to ask is, what is the number of events in an interval capital T? So usually we will denote the timing of events in using lowercase and intervals using capital case. So, for example, if we believe that this Georgopoulos neuron is a Poisson neuron, what would be the distribution of number of spikes in 1 second? What do we expect from a Poisson, from a homogeneous Poisson model? So the way to address this question, let's say that this is our interval T. So what we will do, we are going to divide this interval T into an infinite number of delta T's. So the number, let's do it here. So we will consider each one of these intervals separately. So the number of intervals we will denote it by capital N, will be equal to T over delta T. This will be the interval, total interval divided by these delta t. And now we can ask the question, what's the probability of k events? So what's the probability of k events? Exactly k events. So everything is in the limit of delta t going to 0. So what's the probability of k events? How are we going to compute it? So what's the probability of an event taking place here? It used to be written somewhere. Lambda delta t. So the probability of event in interval delta t is p is equal to lambda delta t. So what's the probability of k events? So in order to obtain exactly k events, we should have exactly p events here. The probability would be p to the power of k. So this is p to the power of k times 1 minus p to the power of n minus k. So this is the probability that if an event did not take place in all other n minus k cases, times what? The number of different configurations. I'm sorry? Okay, times the number of different configurations. I mean, it could be that all events took place here and none took place here, or it could be that all of them took place here and none took place here, or any other combination. How many combinations do we have? N choose k, n over k. So this is the probability of k events. So just writing it in a different way, this is the limit of n going to infinity of p would be lambda t over n to the power of k times 1 minus lambda t over n to the power of n minus k times n factorial divided by k factorial n minus k factorial. Right? This is lambda delta t, and this is 1 minus lambda delta t. And we're interested in the limit of n going to infinity. So let's just rewrite it in a different way. So this is equal to the limit n going to infinity of lambda t to the power of k, taking this 1 minus lambda t over n to the power of n. I'm taking this term times 1 over k factorial times n factorial divided by n to the power of k n minus k factorial times 1 minus lambda t over n to the power of minus k. This is just rewriting these terms. So this came from this came from this. The n to the power of k is here. The 1 minus lambda t to the power of minus k is here. And here I have the n factorial divided by n minus k factorial, and the k factorial is here. Now the reason that I wrote it this way is the following. in the limit of n going to infinity, what will happen to this term? What n goes to infinity, so this will go to 1. What about this term? So n factorial divided by n minus k factorial is equal to n times n minus 1 times n minus 2 times up to n minus k. We have k, n minus k plus 1, so we have k elements divided by n to the power of k. So this will also go to 1, right? What will happen to this k, to this term? The limit of n going to infinity of 1 minus lambda t to the power 1 minus lambda t over n to the power of n, e to the power of minus lambda t. So this would be equal to lambda t to the power of k, e to the minus lambda t divided by k factorial, and this is known as Poisson distribution. So this is a relationship between Poisson distribution, which I'm sure you all encountered in one of the probability and statistics courses that you took along the way, and the Poisson process. So this is a Poisson process, and this is Poisson distribution. And Poisson distribution is just a number of events in a homogeneous Poisson process. We are out. Unfortunately, we are out of time. So on Thursday, we will continue studying the signature of Poisson processes, and then relate them to biological and non-biological processes. Questions? So I'll see you on Thursday.
קשה מכל.
אה?
מי? אני רוצה להגיד שאני לא שתיתי היום. אני חושבת כבר מצפה שבת. אני חושבת שלא. גם אם לא, זה בשישי בצהריים, אני חושבת שזה אם ממש אני חושבת ללכת, אז אני כאילו... שם חזר. כאילו, כי המטרה שלי היא ביום שבת כבר להגיד. כאילו, בשישי מצידי, שנצא. אני אלך קצת, פשוט... כי אין לי אופציה כל כך אחרת. למה התחלת היום? היה לי ממש כזה בטן, כאילו לא הצלחתי את לא, זה לא ש... כאילו אני לא נשארת. סבבה, שיוצא החוצה, אני לא מתביישת, כאילו, בצד שאני ישנה. אבל, כאילו, אני נשכבתי עקוף כזה, על הבטן, כאילו, כוחה, חשות לפעמים. טוב, גם אמרתי, טוב, אני לא יכולה, אני אקלה לתחילה גם קצת ירמה. ורק שאכלתי, כאילו, ביצה, זה ממש מוזר. ביצה? זה גורם לזה לתת? לא, כי כאילו אחרי כל זה היה לי כאילו מין כזה שעה שכאילו הייתה לי מין תחילה וכזה הרגשתי רע, כזה כללי, אתה יודע? זאת אומרת, טוב, לא אכלתי